Okay, so good morning, everybody. Uh, since I will not be here on Monday morning, I have a doctor's appointment. So this is a recorded lecture in continuation to what we were doing last time. And if you recall, I'll take you back to the slide where we had just started to talk about the over-the-counter pharmaceuticals. And if you remember last time, we had also discussed about what these over-the-counter pharmaceuticals are. So these are essentially medicines which are very easily available if you go to a chemist. However, because of sometimes the careless usage of many of these compounds, they end up in the wastewater streams, and these are the new emerging pollutants. So the case which I'm going to be discussing is of what you will see here is pseudoephedrine, which you will see the third one from the bottom here. So this is one of the compounds which I'm going to be talking about, which is identified as an emerging pollutant. And the data on its toxicity is given here. That is, it has been tested on algae, on then you look at the numbers here on Daphnia, and then fish aquatic toxicity, etc. So on the basis of this data which has been generated, we know that it is a pollutant. Now let us see as to what is the origin of this pseudoephedrine, and if there are compounds which are very similar to it, which have now become identified as emerging pollutants, and what kind of methods can we develop in the laboratory in order to look for solutions, biotechnological solutions, in order to break these down. So the paper which I'm going to be discussing is a relatively recent paper, and the title of the paper is Biodegradation of Ephedrine Isomers by a Bacterium, Arthrobacter. It is a new strain which has been found to have potential of degrading ephedrine, pseudoephedrine, by virtue of producing new kind of enzymes which are called as dehydrogenases. Okay, so let me give you a little bit of an introduction on what ephedrine and pseudoephedrine are and where do they come from. So as you see in the first point, that this is like one of the stereoisomers, which is RS ephedrine or SS pseudoephedrine. These are present in various over-the-counter drugs. They are commonly used as decongestants and stimulants. By decongestant, what we mean is that it is a cough syrup, which you can normally, if somebody has a cough, you don't have to go to the doctor to buy this medicine, so you can easily buy a cough syrup, and it contains this both ephedrine as well as pseudoephedrine. In fact, you can take a bottle of it sometimes and have a look at it, and you will see that both of these are the constituents of the cough serum. And stimulant, because it has some kind of a stimulating property also. So some people are also misusing it as a drug because it's sweet and it gives a very heady kind of a feeling. So lately it has also begun to be misused by young people. Now, what happens to these molecules in this cough syrup? These are eliminated by the kidney. They are partially acted down by enzymatic action in the kidney, only partially, and then they are excreted with urine, leaving 70% of this ephedrine and nearly 98% of the other isoform, that is the pseudoephedrine, in a high dose. So in other words, the body is not capable of metabolizing them entirely, but it only carries out one enzymatic step, and the rest of it, okay, 70% of these or 98% of pseudoephedrine, for example, leaves the body, and obviously it finds its way into the wastewater streams. According to the 2017 International Narcotics Control Strategy report, the top 5 world economies, they export more than 1,000 tons of this pseudoephedrine and more than 100 tons of this ephedrine. Okay, so in other words, there is easy access of this all over the world also, because it is in the commercial trade. It's not regulated at all. It can be exported to other countries very easily and used, for formulating many of these cough syrups, etc. And both of these compounds have been demonstrated to have a potential negative impact on many biological systems, which I'll show you in the next slide. But in this slide, what is given is a list of these ephedrine compounds. Okay, so as it says here, the first compound which is here boxed in red, it finds wide application as a bronchodilator to treat bronchospasm, which is associated with, so people who are asthmatic or they have a bronchitis, they have swelling of the windpipe, it is given as a suppressant to those people. Okay, and shortness of breath, etc. That time this particular cough syrups, these are prescribed. So both this, this is a part of this part of the cough syrup, and similarly the second one, which is 1s2s plus, which is shown here again boxed in red. Okay, this is used as a decongestant. All right, now what happens is that these are of course directly released into the wastewater bodies. In addition to that, you will also see these two other isomers which are shown here, which is a 1s2r plus ephedrine and 1R2R minus pseudoephedrine. All right. Now, where do these come from? What happens is that during wastewater treatment, both of these are partially isomerized to the SR ephedrine, which is even more environmentally persistent. Okay, so although these are the two original molecules which are boxed in red, but during wastewater treatment processes, either due to bacterial action or due to other action, both of these are converted into these other isomers which are even more environmentally persistent. That is, in the environment, there is no enzymatic activity, no microbial activity which is able to degrade them or metabolize them completely into harmless compounds. Okay, and it is also classified when you perform the tests on them. It is classified as chronic 2. That is, if you look at the EC50 value in either in the fish, or if you look on, for example, Daphnia test, then the EC50 value, effective concentration at which it causes 50% mortality of Daphnia, is between 1 to 10 milligram per liter level. On the basis of this, it's now classified as a chronic 2 toxicant. Okay, and both of these, okay, apart from the original ones, both of these compounds, they have been found to have a significant toxicological effect on Daphnia, and this is that algae which is a part of that kit which I discussed with you earlier, and also on Tetrahymena. So there is a data which I have taken where these studies have been performed. So what you see here, for example, is these are the isomers here. They have been tested individually for their toxicity using the standard eco tests, which are carried out on either Daphnia magna, and then you have an EC50 value for 24 hours, or EC50 value that is within 48 hours if it is able to cause 50% mortality. Okay. And similarly, this is your algae, for example, and this is Tetrahymena thermophila. So if you look at the data, all these 4 isomers have been found to exhibit toxicity, and these are the corresponding values here. And if you want to look at the raw data, for example, here for Daphnia magna, this is something which I had shown to you in one of the earlier lectures also, that on the x-axis you are plotting log to the base 10 concentration, and then you have like the red circles here is your percent mortality data, which is measured in 48 hours by replicate 1 and replicate 2. What we mean is that the experiment was carried out in duplicate. All right, so either the red circle or the red diagonal here, they represent results of the duplicate experiments. And what you see in blue here, for example, are again the duplicate, but a 24-hour data. So you begin to see a sigmoidal curve, for example, here. All right. So, one can always calculate what is the EC50 value. So wherever you see a 50% mortality, you can just go back, and for both the blue line and also for the red graph, you can plot now what is the concentration at which you are getting 50% mortality. So that is the EC50 value which is given in this table. Okay, and I also expect that you should be able to plot the graph. If you are given the raw data, then you should be able to plot. So in other words, ephedrine and pseudoephedrine have been already proven. They have been shown to have this kind of a toxic effect. Similarly, the raw data I'm sharing with you here is for Tetrahymena thermophila. Again, concentration is on the x-axis and percent inhibition of growth is on the y-axis, a typical sigmoidal type of a dose response curve where you can measure the EC50 values. Okay, so you have, wherever you see a 50% inhibition, you go back and you find out what is the concentration at which you are getting 50% inhibition. So now, um, if we look at, for example, now the paper which I am discussing has tried to first put down, for example, what is already known about how ephedrine and pseudoephedrine are metabolized in mammalian kidneys or in some other bacteria which had been reported till then. Till that paper came out, there were already some studies which had been carried out. Okay, so in mammals, how does it get metabolized? Partially, as I said, not completely to carbon dioxide and water, but partially. Okay, so what happens in a mammalian system is that you start out with ephedrine right here in the center. Okay, and there are two kinds of activities which can occur. The first is oxygenation, which is carried out by what is called as, which is abbreviated as O, and this activity of oxygenation of ephedrine is carried out by cytochrome P450 monooxygenase. Okay, so we will see that either in many mammals or in fungi, for example, one of the first steps which is used in terms of breaking down aromatic compounds is addition of an oxygen atom, or it could be addition of two oxygen atoms. So those enzymes which add only a single atom of oxygen are called as monooxygenases. Okay, so in this structure, what you see as you go from ephedrine to hydroxyephedrine, one of the hydrogens here, for example, now it becomes a hydroxyl group. So there has been addition of a single oxygen. Reaction is catalyzed by a monooxygenase, which is then followed by the second reaction, which is called as the D1, which is actually a dehydrogenase reaction. And what happens is that right here you see, so this is now reduced to an NH2 group. The resulting molecule is a parahydroxy ephedrine. So this is as much as it can be metabolized on this stream. Or you may have first a dehydrogenation, which is then followed by oxygenation. So it is exactly the same two reactions, except that the sequence is going to be altered. And you have the formation of hydroxy norepinephrine. This is excreted by kidney. So as I said, there is only a very limited amount of metabolism of this compound in the human kidneys, in the mammalian kidneys, and it is excreted. In Arthrobacter, which is a bacterium, till this paper came out, there were bacteria which had been reported, and one genus is Arthrobacter globiformis. And what it does is that it takes ephedrine or it takes pseudoephedrine with the help of enzymes which are NAD dependent. These enzymes are called as either E1, again E1, which is NAD dependent enzyme, which carries out a dehydrogenation. So you see this amine group here, NH group is converted into an imino group, like you see here. So from both ephedrine or pseudoephedrine, you have a resulting methyl imino phenyl propanol, which is then quickly hydrolyzed because the amino groups are very, very unstable in water. It immediately undergoes hydrolysis and you get phenylacetyl carbinol. Okay, so this was the pathway which was known till that paper came out. So what were the objectives which were set out in this paper? First was, can we find a biotechnological route? Can we find microbial systems which are capable of breaking down ephedrine and pseudoephedrine so that, such kind of microbial activities or enzymes could be purified on large scale, and then they can be applied to the treatment plants where they can break down both ephedrine as well as pseudoephedrine. So the strategy was to identify a bacterium that can use this as a sole source of carbon. Okay, and generally what is done is that, for example, in order to do that, a soil sample can be collected, and then the soil sample is now diluted out. For example, you can do a serial dilution of that, and you just take it in a test tube which contains only ephedrine. Okay, or you may set up a parallel tube in which you will have ephedrine plus you will add a nitrogen source, so on and so forth. Okay, so the idea is that if there is any microbe in this soil consortium which is capable of using this ephedrine or pseudoephedrine as a sole source of carbon and nitrogen, then it'll be capable of growing in the first tube. So whatever grows in that tube can be taken and it can be plated out onto an LB medium. All right. In the second tube, as I said, that you have ephedrine plus you are adding an external nitrogen source, because ephedrine, as you will see, okay, it already contains nitrogen. Okay, so the question is that sometimes you may want to have a bacterium which can use this both as a carbon source as well as a nitrogen source. Okay, however, many microbes may use ephedrine as a carbon source, but then a nitrogen source like ammonium nitrate or ammonium chloride may have to be supplied from outside. Okay, so in the second tube, which I just mentioned, is that you add an external nitrogen source in order to select for those microorganisms which can use ephedrine as a carbon source, but they can't use it as a nitrogen source. And that is why they are dependent on an externally added nitrogen salt. Okay. So keeping that in mind, all right. So the strategy now was to identify a bacterium from a soil mixture which could use this ephedrine as a sole source of carbon. So in this process, when they did this kind of an experimental setup, there was one bacterium which started to grow on ephedrine when nitrogen was supplied, and that was now called as Arthrobacter TS15. It was a gram-positive bacterium. It was an aerobic bacterium. Okay, and just is the case always that whenever you isolate a new bacterium, you send it for 16S ribosomal RNA sequencing. Okay, because you want to identify what kind of a bacterium it is, you want to place it in a genus or in a species. So on the basis of this 16S ribosomal DNA sequencing, it was found to closely be related to a previously reported Arthrobacter, but the strain number was as is given here. It was also found to have 16S ribosomal DNA sequence which matched with this is Orobasidium oriscens TC1 and an Arthrobacter species strain, so on and so forth. Okay, so it was established that the new bacterium which is isolated was an Arthrobacter. Okay, so once the experimental results indicated that in the original consortium you had a bacterium, then you can always, you can streak it out for single colony. The colony is purified, you have identified, and now what you do, you set up in a test tube experiments where you are going to give any one of these isomers of ephedrine, like the RS ephedrine or the SR ephedrine or pseudoephedrine or pseudoephedrine as a sole source of carbon, plus a nitrogen source was added into these test tubes. Okay, so the data which is shown here indicates that as the bacteria is incubated in a medium which contains only ephedrine and a nitrogen source, okay, the bacteria is able to grow. So what you see in the solid circle here, if you quickly go and look over here, okay, it is estimation of the culture growth. All right, so culture growth is monitored by just measuring optical density at 600 nanometer. Okay, so you see on the y-axis here OD 600. So the black dots here represent that after the culture, this single isolated Arthrobacter is inoculated into this medium, it starts to grow. All right, and this is the increase in the cell OD as a consequence of growth. All right, what is also measured is, for example, the degradation of ephedrine, because ephedrine is something which can be measured by HPLC. So all the samples which have been periodically removed when the bacterium was growing on this, they are also taken and you measure what is the concentration of ephedrine. And what you see that at time 0, you start out with concentration 5 millimolar in the growth medium, but then gradually you see that there is a decrease in the concentration, and eventually in about 50 hours time or in about 70 hours time, the entire ephedrine which was in the growth medium is completely metabolized. Okay. In addition to that, the triangles, they represent another compound which is methcathinone. Okay. So I will tell you what is the significance. So this way, the bacterial inoculum into a medium which doesn't contain, any carbon source other than ephedrine, it only contained nitrogen source in addition to that. Okay, so on all these four isomers, the ones which are in red are the naturally occurring ones, and the ones which are shown, which are not circled by red box, are the ones which during wastewater treatment, as I said, there is a microbial activity which can convert this isomer into this form. So these two are those ones which are resulting as a result of this passing it through the wastewater streams. So the results in these figures, they illustrate that the pseudoephedrine and ephedrine, for example, you have the data for ephedrine right here, you have the data for pseudoephedrine here, all right. So all of these are metabolized right here, you see, and there is an increase in the cell OD. So this particular bacterium is able to use this particular ephedrine, okay. And from all the four graphs, you can also measure what is the doubling time, because the cell OD, it goes in a certain period of time during the log phase. You know how many doublings it has gone, so you can measure, for example, what is the doubling time, or the same cell OD data can be converted. It can be plotted on a semi-log plot. You can convert it into a log plot and measure versus time, and the slope of that will give you the growth rate, etc. So what you find from the data is that SS pseudoephedrine is the one which is most rapidly taken up by the cell, okay? And half consumption, that is the time taken to metabolize 50% of the externally added pseudoephedrine, was 4.4 hours. So it is pretty rapidly metabolized by the cells, confirming that this particular bacterium is capable of metabolizing both ephedrine as well as pseudoephedrine. Now, what is shown in this particular picture is that there are several standards, like for example, ephedrine, pseudoephedrine. You would like first of all to estimate them by gas chromatography, by MS, to know what is their retention time, because if I need to estimate many of these compounds, I need to have a method, which in this case is gas chromatography. Okay, by gas chromatography, what we mean is that the compound which you are taking, like ephedrine or pseudoephedrine, is converted into a volatile phase. And just as you do liquid chromatography, you can do gas chromatography also. Now, the vapors of ephedrine and pseudoephedrine are separated, for example, by a two-phase system, where you have a liquid stationary phase, and then you have a gaseous mobile phase, which separates these compounds by what is called as a GC chromatography, which is a gas chromatography essentially. Because unlike the normal chromatography where the mobile phase is a liquid, in the GC you have the gas as the mobile phase. Okay, so now what we see in panel A here is, for example, phenyl propane 1,2-dione is a standard compound, and under the experimental conditions which have been standardized in this particular, in the laboratory, in this paper, you find that it gives a single peak, and there is a certain M by Z value at which it appears, and in the panel which is just given below this. All right. So if this particular peak is taken and now it is injected into a mass spectrometer, then this molecule, which is actually the phenyl propane 1,2-dione, undergoes fragmentation. Okay, because there is only as many ways that this molecule, depending on the bond energies between different atoms, there is only a specific way that the molecule is going to be fragmented. So let us assume that in this particular molecule, this is the first bond to be broken, as a result of which, okay, the entire molecule is going to be shortened by oxygen, by a carbon, which is 12, oxygen, which is 16, and then there could be a methyl group here. Okay, so CH3 plus a carbon, which is actually a CH here, and plus an oxygen. If you add them up, those numbers, okay, so a methyl group is carbon is 12 plus hydrogen 3, that is 15, plus CO, which is another CH, which is 12 plus 1, 13, plus an oxygen, plus 16. So you should expect to see another fragment here. This could be the original molecule. Then another fragment is going to appear here, which will be lesser by this particular moiety right here. Then you could also break down it here. You could break down, like, finally you have a benzoic, you could have benzoic acid, for example, eventually. So in other words, all of these, if the original molecule is this, okay, every time when it is subjected to fragmentation in a mass spectrometer, there is a very characteristic fingerprint it is going to give. That means the fragments which are going to be generated will always be unique, each one of them having a specific molecular weight. So such a pattern is called as a fingerprint for propane, for phenyl propane 1,2-dione. Similarly, for this molecule, which is actually, you will recognize it is benzoic acid. If you take it into a mass spectrometer, there is only a typical fingerprint which it is going to give you. Okay, so in other words, what it allows you to do is that in your culture broth, because ephedrine is getting metabolized, there are certain compounds which are going to be formed. And these compounds can be measured by gas chromatography. And the identity of these compounds can be further established by doing a mass spectrometry. So in other words, it allows you to identify that particular compound. So this was already carried out in this investigation. So different molecules were taken based on the data which they had got, which you will see in the next slide. So what is shown in this particular slide here is that now, you have those cultures which you just saw, and this bacterium is growing on ephedrine or on pseudoephedrine. So what is taken is that the culture filtrate is taken, the cells are centrifuged out, okay, and now a GC chromatogram is prepared. So this cell-free culture supernatant is taken and it is injected into a GC, not GCMS, but just GC. Okay, and you will get several peaks, each one of them corresponding to the molecules which are present in the culture broth. Okay, so you have, for example, there is a peak which is appearing at position 1, then right adjacent to that is peak 2, then there is a peak 3, all right, and all of them have a certain, this is given in the retention time in the gas chromatography. Retention time, why? Because in liquid chromatography, the time that it elutes out of the column under that chromatography step, okay, is usually measured by the time. So molecules which are going to elute out first will have a much shorter retention time. The molecules which have a much higher affinity for the stationary phase, they will stay longer and they will elute out of the column much later. So their retention times are also going to be much longer. Okay, so that is how these are 18 minutes, or these are like 21 minutes, 24 minutes, so on and so forth. So 1, 2, 3, 4, 5. So at least 5 different molecules were identified. And when each one of them is further taken into a mass spectrometer, as I said. Okay, so in a GCMS, this particular peak can be selected. It can be made to pass through your liquid chromatography tubing, and it can be injected into a mass spectrometer. And then you try to see by, these are the fingerprints because it is now broken down. And this fingerprint that you see here will allow you to identify as to what this peak at 1 was. Okay, so the peak at 2, by virtue of having a specific retention time and by virtue of displaying this kind of a fingerprint, is now concluded to be benzoic acid. All right, so I hope it is clear to everybody. And please, when you listen to the lecture, if you have any doubts or something, we can always come back. So in other words, all these molecules which are now identified in the culture filtrate, okay, they were identified by way of a mass spectrometer. So you could say that these are the different molecules which are resulting because of metabolism of both ephedrine as well as pseudoephedrine. Okay, so 5, you see right here, 5 is ephedrine. All right, so ephedrine has a structure like this, and this is the kind of a fingerprint you have. So the peak which is appearing at 5 is actually the substrate, the initial substrate, and all of these are the resulting products from ephedrine. So now, so it is already identified that, these are the metabolites. But then the second question which was put down in the study was, how do we really identify what kind of an enzyme, extracellular enzyme, is secreted into the growth medium? Because eventually the objective of every researcher is to be able to focus to identify which enzyme is actually responsible for breaking down ephedrine, so that tomorrow many of these enzymes, the genes which are coding for these enzymes, can be cloned and they can be produced in large quantities, and then they can be put to some kind of a commercial usage. Okay, like in wastewater treatment plant or wherever you expect that this pollutant is there, then you can always do that. So how did they go about enriching this particular enzyme in the culture filtrate? So the bacterial culture was grown, and then culture was supplemented with a very small amount of this pseudoephedrine to induce oxidizing activity just 1 hour before harvest. Okay, so although initially, it takes time and it grows, but maybe by the end of the culture growth, there was not enough of, or maybe the oxidizing activity is completely used up. So there is a pulse of pseudoephedrine which is given just 1 hour before the harvest. And this is so that when you give this pulse, again, there is an induction of that particular enzyme so that now it can be isolated. Then, so the entire culture filtrate is taken and enzyme is purified. And basically at every stage, what you are doing is that you are enriching for an enzyme activity which is able to break down either ephedrine or pseudoephedrine. Okay, so for any enzyme, during the enrichment procedure, during purification, you always start with the substrate. So in this case, their substrate which was used was ephedrine or pseudoephedrine. So as they purified, they were trying to enrich for this particular activity, and so it was purified, and the band was isolated from the STS page gel, and it was subjected to tandem MS peptide fingerprinting. So, those bands which you see now on the STS page gel, they're already stained blue. So when you read the paper, you will see that the band can be taken out. It can be subjected to trypsin digestion, and all the different peptide fragments which are made there, they are passed through a liquid chromatography system, and then there is a fingerprint which is given. Okay, that this particular fragment, which could be long, it has a size of, let's say, 1500 Dalton, and then 1500 means about 12 to 15 amino acids. Okay, so if there is a sequence of amino acids which is of this nature, okay, then it's likely to have a peptide fragment mass of 1500 Dalton. So on the basis of that, these peptide fragments were identified. Okay, and then the gene was identified. So let me quickly show you that what were the results of this peptide fingerprint. Okay, so I hope you will be able to see this. And so in this table S2, if you see, so it is like this particular peptide, the protein which is now purified, which has been enriched in the culture, is subjected to MS peptide fingerprint. It is subjected to trypsin digestion, and the peptides will when you subject them to a search, you will find there were 55 hits. In other words, so in the database, the sequences could match against enzymes like catechol dioxygenase, which was from Ralstonia, or a bifunctional. So there were approximately 51 hits. Okay, amongst them, what they also found, that it also matched, like one of the peptide fragments matched with a putative short chain dehydrogenase from an uncultured bacterium. Okay, and the mass of it was, and although the probability was very low, only 55. So if you see, what does the last column represent? Score. Generally, the higher the score, the better, or the more sure you can be that this particular fragment which you have isolated is going to match that particular enzyme. But apart from all this, they also identified that there was a match with a dehydrogenase, with a low probability. Okay, so in this particular cases, what is done is that that sequence is taken, because now you know that there is a dehydrogenase. Okay, so those DNA sequences which are already available in the genome are searched for the presence of these sequences, and that is called as genome mining. Because what was done by the investigators was that the bacterium which they had isolated was subjected to whole genome sequencing. So you knew exactly what are the nucleotides in there, what is the genetic makeup of this bacterium, and now you try to see, for example, that this short chain dehydrogenase with which it showed a match, you tried to see what is the corresponding nucleotide sequence of this in that uncultured bacterium, and whether such a sequence can be identified in your newly isolated bacterium. Okay, so that is called as a rational genome mining. When that was done, there was a fragment of DNA identified in this newly isolated bacterium. Okay, so let me show you exactly. Just excuse me, we will go back to our, okay. So now in this slide, all right, so what you are able to see, for example, that when the nucleotide sequence is subjected to this, there is a match which is found, all right? So the nucleotide sequence of the dehydrogenase was mapped to one fragment, which was in this new bacterium, Arthrobacter species TS15, okay? And it belonged to, for example, so that sequence matched somewhere here, and that DNA sequence also matched with a part of it here. And both these were actually a part of a much larger operon, which is called as a catechol operon. Okay. Now, in Arthrobacter species, this was the newly isolated bacterium for which the genome sequence was determined. Now, when the entire genome was compared with the other Arthrobacter, which was already available in the database, okay, the first operon which you see here, which is the CAT1 operon in this new bacterium, was also found to be present in this Arthrobacterium oriscens, which was already available. However, this particular bacterium was not able to metabolize pseudoephedrine, whereas Arthrobacter species TS15, which is the newly isolated bacterium in this species, was found to metabolize. Okay, so over and above this, it was also found to contain, this new bacterium, another operon which contained many genes which were common to what was found here. But over and above that, there were also several new genes which were identified in this operon. And this was called as the CAT2 operon. CAT stands for catechol metabolism. Okay, so many bacteria are able to break down catechol, etc. Okay, so it's able to do this. So on the basis of this, so now the question is that you have been able to locate it in the genome sequence, and tentatively these were called as ephedrine dehydrogenases, and they were called as pseudoephedrine dehydrogenases. Because the genome sequence is available with you, you can have primers on both the sides, and you can take genomic DNA as the template. You can amplify out these two genes, and that is what exactly was done in this study. You can read the remaining details here, but essentially what is done is that the genomic DNA of this new bacterium was taken, the primers were raised, and EDH was amplified out. Pseudoephedrine dehydrogenase was amplified out, the gene which is encoding for them, and it was cloned into E. coli. Okay, and now the E. coli expresses this enzyme. So cloning of both the genes was carried out, its expression was carried out, and it was purified from E. coli. And again, its activity was monitored on RS ephedrine and RR pseudoephedrine. And what was found is that this ephedrine dehydrogenase, which is now isolated, cloned, the recombinant EDH exhibited strict enantioselectivity for the oxidation of ephedrine and RR pseudoephedrine. Similarly, the other gene, when it was expressed in the bacterium, the purified enzyme exhibited stereospecificity for SS pseudoephedrine and SR ephedrine. Okay, and the data is also given on the activity of the cloned enzymes, which is in figure S10 in the supplementary data, which accompanies that particular paper. So in other words, both of these enzymes were now identified in this bacterium, and a pathway was therefore put down, because unlike what was shown in Arthrobacter, that there was a different pathway which was followed. But in this particular bacterium, meth-cathinone was already identified in the culture filtrate, which had not been identified by the earlier bacterium. All right. So therefore, what was proposed down based on the data which was obtained with the cloned enzymes is that Rn, the pseudoephedrine, for example, is acted upon by ephedrine dehydrogenase to give rise to methcathinone. And similarly, pseudoephedrine is acted upon by the pseudoephedrine dehydrogenase. It is an NAD plus dependent enzyme, and you get methcathinone. So in other words, there was a new pathway which was present in this newly isolated bacterium, which was capable of degrading ephedrine. Okay, so in other words, the question which you were trying to ask to begin with is that is there going to be a microbiological route? Is there a biological route by means of which these emerging pollutants can be metabolized? Okay. So on the basis of this, for example, the degradation pathway was put down. So what was put down is that, for example, you start out with ephedrine, and the first enzyme, both ephedrine dehydrogenase or pseudoephedrine dehydrogenase, is capable of acting on them. And the first molecule is methcathinone, which is also identified in the culture filtrate. However, it's not very stable, and it immediately loses its amino group, and what is formed here is phenylpropane 1,2-dione. So if you recall quickly, in the culture filtrate, this was also one of the metabolites which was detected. Okay, so go back to one of the earlier slides where through mass spectrometry, the culture filtrate was analyzed and phenylpropane dione was identified also as one of the intermediates in there. So therefore, in this present paper, what is put down is that how does phenylpropanedione or PPD, how does it appear in the culture supernatant is because this methcathinone is highly unstable. It loses an amino group and it can make PPD, which is again acted upon by either ephedrine dehydrogenase or pseudoephedrine in order to give rise to phenylacetyl carbinol or PAC, which is also detected in the culture filtrate. Okay, and eventually it loses this and you get benzyl dehyde is formed and finally benzoic acid. Okay, so in this particular bacterium, therefore, by virtue of having these novel two enzymatic activities, it is able to break down either ephedrine or pseudoephedrine into benzoic acid. And incidentally, many of the soil bacteria, okay, are able to use benzoic acid as a carbon source. Okay, just as any other acid, like many microbes will use acetic acid, or they can use formic acid, they can use other propionic acid, etc. as a carbon source. Similarly, many bacteria are able to degrade these acids, aromatic acids also like benzoic acid. Okay, so therefore this was now put down as a pathway. And so eventually, what we have learned under ecotoxicology is that what does the subject matter deal with? You saw the various standard tests which are available, the dose response curve and the standard tests which are available, and they are adopted by all the laboratories, EC50 values. So standard microbes are taken so that everybody can report their results in terms of something which can be compared across the laboratories. So we talked about different tests, and we also looked at the case study of an emerging pollutant like ephedrine. And you also learned about what kind of methods are used currently to isolate organisms which can decontaminate such toxic compounds. Okay, so with this, the subject matter of ecotoxicology is finished. And what I'm going to do very quickly in the next maybe 10 minutes or something is now ecotoxicology is over, and what we are going to start next is our discussion on microbial biosensors. Okay, so I'm just going to take a break for about 5 minutes and then we will come back. So 5 minutes we will take a break and then I'll come back with the lecture. Okay, so let's start with the next topic, that is on microbial biosensors. And so I will just introduce the topic to you, and then when we come back, we will do case studies on this. So the question is that what is the need for a sensor when you talk about environmental biotechnology? All right. So the need for a sensor is that you would like to rapidly detect as well as quantify pollutants which are present in the environmental samples, because it is very necessary for human health. It could be a river, which is eventually going to be a river water, which is going to be eventually supplying, let's say, the drinking water to households, and you would like to know how much of a given pollutant is there. All right. So for that, you need a sensor. Or the sites which are suspected for the presence of the toxic compound contamination are traditionally, so in general what has been happening till now is that many of these samples, there is no quick sensor which is available to detect the presence of a pollutant. But what is normally done is that, as it says in the second point, that the sites which are suspected of the presence of a toxic compound are traditionally, they are analyzed and quantified by a number of analytical methods. Okay, so you will take a sample, you will bring it to the laboratory, and you will find the presence of, let's say, if these are metals or certain other things. You will do an analysis by atomic absorption spectroscopy, or by mass spectrometry, by emission spectroscopy, or by gas chromatography. All right. In other words, that these methods are extremely expensive. These are time consuming. They also require transport of the sample from the site, from the actual site of contamination to the laboratory. And they do not tell us about their bioavailability. That is, that even if a pollutant is really present there, okay, the question is that how much is really going to enter into a biological system. That is what we mean by bioavailability. So just the mere presence of a pollutant in the environment doesn't mean that it is also bioaccumulating or it is available to the biological system. None of these. Okay. So, and the last point, in addition, these technologies may not be accessible worldwide, particularly in the developing countries. So all of these methods which we have described here are very, very sophisticated analytical methods, and not everybody has an access to that. So therefore, there is a need for uh, biosensors, which could be very quick, and we will see that biosensors can be of different type. All right, and they don't necessarily involve atomic absorption spectroscopy or very, very, highly sophisticated instrumentation. So some examples which I'm going to give you now. So what we are trying to say is that the sensors which are going to be using, which you would want to use, there should be biological system, biology based system. Okay, so these biosensors, in other words, are analytical devices which have a biological component, and they also have a transducing element for the detection of a specific compound. So what is the biological component? It could be either enzyme, it could be an antibody, it could be living cells, or it could be immobilized enzymes. And I'll explain it to you in this particular picture. Or it could be, for example, so these are biological sensors are these are some of them, like enzymes, antibodies, etc., are expensive, whereas the microbial biosensors are not very expensive, in which you're going to use whole microbes for a given purpose. Okay. So although they are expensive, but these are sensitive, and sometimes they're not always ideal for field applications like enzymes, etc. But right now, I think in the broad definition of a biosensor, we are trying to look at either the whole cells themselves or the components thereof, which can serve as a sensor. Okay, so one example to illustrate that point before we come to okay, before I give you an example, let me just explain as to what a biological sensor does. So we said it could be something like an enzyme. So you have, for example, so in a sensor, there is going to be a bioreceptor which has the capability of binding to that particular analyte. Okay, so it is an enzyme here. For example, it could be glucose oxidase, and you are trying to detect the presence of glucose, like in the blood or in the glucometer, which you normally use at home. What you have is that there is glucose oxidase, which is immobilized on a strip, and what it detects is the presence of glucose in the material which you put, which is a drop of blood on that. All right. And it's highly specific because glucose oxidase, out of many components which are present here, will only be able to bind to glucose. So it binds to glucose, okay, and as a result of which there is a chemical reaction which is going to occur, and electrons are going to be released, etc. So that particular thing is converted. Any kind of a response which you have could be either electroactive substance, it could be electrons which are released, or there could be a pH change, there could be protons which are released as a consequence of this binding, or there could be heat which is generated, there could be light which is generated, or there could be a change in the mass. So any one of these is going to be now changed into an electrical signal by means of a transducer. So that is the transducer component of a biosensor. So a biological sensor has a receptor or a bioreceptor, and it also has a transducer. So here is a list of what all can serve as a receptor. As I said, it could be an enzyme like glucose oxidase, or it could also be an antibody. Okay, and you are checking, for example, in the blood. Sometimes, people who have cancer, they have special type of antibodies which are circulating in the blood, and you could choose an antibody which is highly specific to detect that particular antigen in the bloodstream. So the binding between antigen and antibody takes place, so you can detect, for example, so in this case that particular antibody becomes an analyte. Okay, and the antibody which you have now on you have a receptor, it binds to that. Okay, as a result of which there could be a change, there could be a pH change, there could be a change in the heat when antigen and antibody bind. It's a chemical reaction which can then be translated into some kind of an electronic signal. All of these options are given to you here. Okay, so eventually all of them will be able to convert it into an electrical signal which can be picked up by the detector. So that is what basically a biosensor is going to do. So piezoelectric, for example, measures change in the mass. Electromechanical measures a change in the electric distribution. So the properties of the different transducers is given here. Okay. So I'll give you one example because it is one of the most commonly used biosensors, and it's based on using glucose oxidase system. Okay, so in the green, what is shown to you here is glucose oxidase, which can be immobilized on a paper strip. So this glucose oxidase is very specific, and it recognizes glucose as a substrate. It converts it into gluconic acid. Okay, but in the process, during the oxidation process, oxygen is taken up, hydrogens are collected, and it is converted into hydrogen peroxide. Okay, or it could be any oxidized mediator which is converted into a reduced mediator. All right, and these electrons, for example, can be detected in the electrode, which is now going to measure the detection of electrons. So you can always convert that signal into a change in the signal in the electrode signal, and that tells you how much of glucose is present depending on the response which you get. Okay, so that is what basically an enzyme sensor is going to do. Similarly, for the detection of urea, so on and so forth. So the stress is also going to be that while both enzymes and antibodies can be used as sensors, what about using the whole microbes as biological sensors? Okay, because many of the case studies which I'm going to be describing to you will show you how the entire microbe can be used as a sensor for detecting the presence of certain pollutants in the soil or in the water. Okay, because cultivation of microbes is a lot more easier. You don't have to purify any enzyme, and microbial sensors can be very versatile. They are not only like some points are listed here, so let me just summarize these with you. That Enzymes and antibodies, for example, these are highly specific and they are selective and they are not general in nature. So if you talk about enzyme like glucose oxidase, it's only specific for glucose, not for fructose, not for any other sugar. Or antibodies are going to be very specific when they are used as biosensor. It's very specific for that particular antigen. On the other hand, if you use microbial sensors, these can detect a wide range of chemical substances because microbes have a versatile biochemical system in there. They can metabolize a lot of compounds, so you can use them to pick up or to detect many other compounds if we use their functions as a sensor. These are very expensive to obtain because there is a high cost of purification preparation involved. However, microbial sensors are easy to popularize. That is, you can easily cultivate them and you don't need to purify these. And these are very sensitive to experimental conditions because enzymes are very finicky, specific pH, specific temperature, so on and so forth. So they are not very robust. On the other hand, the microbial sensors are more tolerant of environmental conditions. There is a certain zone on which they can grow and they cannot grow. And similarly, they are enzymes, as we said, are very sensitive to experimental conditions. These are not multi-purpose like urea, glucose, lactic acid, etc. But microbial sensors can be multi-purpose catalyst, especially when participation of a number of enzymes in sequence is required. So if there is something which you want to detect, let's say you want to detect benzoic acid, okay, then the benzoic acid, the presence could be converted into a fluorescent signal. However, there is a battery of enzymes which is involved in the process. Okay, and that is something which is possible using a microbe because microbes already contain so many different enzymes. So if you develop a system, then all that enzyme machinery is supplied in a microbial sensor. And some other points are also listed here. So I think I'm going to stop here because then it's a little bit intricate, so we will deal with this part in the class. Okay.